Let's talk about anaerobic respiration. Anaerobic respiration occurs in the absence of oxygen. So we know from learning about the electron transport chain that when oxygen is not present in the mitochondria to act as the terminal electron acceptor, the electron carriers in the electron transport chain become clogged up with electrons. This means that they are unable to receive new electrons from the hydrogen carriers, reduced NAD and reduced FAD. So the reduced NAD and reduced FAD can't be oxidized, they can't return back to glycolysis, the link reaction, the Krebs cycle, all of those things stop. So if that was just the end of it, we would be unable to generate any more ATP and we would die. Or at the very least that cell would die because ATP is contained within single cells. And it's in this closed loop cycle. So we have got to find a way of keeping one of those pathways going. And the pathway that we try and keep going is glycolysis. So anaerobic respiration is glycolysis plus a secondary pathway which regenerates oxidized NAD so that that NAD can go back into glycolysis and allow the formation of those two ATP molecules that we know come out of glycolysis. So instead of being able to generate 38 ATP as we would be able to do if we were carry on with the whole aerobic respiratory pathway, we just keep plugging out those two ATPs for as long as we can. And instead of making the pyruvate, which would then enter the matrix of the mitochondria and undergo the next pathway, that pyruvate is converted into something else, depending on if we are discussing mammalian anaerobic respiration or we are discussing alcoholic fermentation, which is anaerobic respiration in yeast cells and in plants. So we're going to talk about those slightly separately. But let's first have a look at anaerobic respiration in mammals. So we've got our glucose, our 6-carbon compound, and that undergoes glycolysis. We know this 10-step reaction, and it produces 2 molecules of the 3-carbon pyruvate. And we know that along the way, it produces 2 NADHs, and it produces 2 ATPs. And those 2 ATPs can go off and be useful to the cell. Those NADHs are unable to go to the electron transport chain. So those NADHs are oxidized in the lactate pathway. So those 2 pyruvate molecules are converted to 2 lactate molecules. Sometimes you'll see this referred to as lactic acid, but they're just the same thing. And the output is 2 oxidized NAD molecules. They re-enter glycolysis and they allow the glycolysis pathway to occur again. And this just happens over and over again, and we produce molecules of lactic acid. Lactic acid first builds up in your cells, then it enters your bloodstream, where it is transported to the liver. Your liver is then involved in oxidizing the lactic acid to pyruvate. This reaction is catalyzed by the enzyme lactate dehydrogenase. So the enzyme uses the electrons that were originally removed from lactate to reduce the molecule of NAD to NADH. That pyruvate then enters the mitochondria in the liver, and then it just enters the respiration pathway. So all that energy that was trapped in the lactic acid isn't lost forever. We get it out in the end, just a little bit later on. Although you absolutely don't need to know this for the A-level spec, this is actually called the Cori cycle. So if you went on to learn about physiology or metabolism in a bit more detail, you might learn about this. So this pathway is overall less energy efficient than the aerobic respiration pathway, but it occurs in extreme situations when a cell or a group of cells doesn't have access to enough oxygen to keep going. When you learn about muscles a little bit later on in the course, you'll also learn about a molecule called creatine phosphate, which is another way in which our muscle cells enable themselves to keep going even when they've run out of oxygen. So that's anaerobic respiration in mammals. You've got glycolysis and then the lactate pathway, so the oxidization of pyruvate into lactate, and then the later conversion in the liver of that lactate via lactate dehydrogenase to produce pyruvate again. Let's just end this section by looking at the equation for anaerobic respiration. Glucose gives us lactic acid plus a small amount of energy, about 2 ATP. Okay, so that's anaerobic respiration in mammals. Let's move on to anaerobic respiration in yeast and plant cells, so alcoholic fermentation in yeast and plant cells. So the equation for this is glucose gives us ethanol plus carbon dioxide plus a little bit of energy. So we can see already that it's quite different. But the principle is the same. They're trying to achieve the same end, which is to generate a little bit of ATP to keep them going, to regenerate the oxidized NAD so that it can re-enter glycolysis and allow it to continue. It's just that instead of doing it in a one-step reaction and producing lactate, it does it in a two-step reaction and produces first ethanal and then ethanol. So let's have a look at that. So we can see that our 6-carbon glucose molecule is broken down into two 3-carbon pyruvate molecules, and along the way we make 2 molecules of NADH and 2 molecules of ATP. This pyruvate is then first converted into ethanal, which involves a decarboxylation reaction and the release of 2 carbon dioxide molecules, and then the ethanal is converted into ethanol. When the ethanal is converted into ethanol, the reduced NAD is oxidized to produce NAD+, which can re-enter the glycolysis pathway and allow it to continue. The ethanol is then excreted out of the cell and lost from the organism. So unlike lactate, it's not reconverted back into a useful substance. But humans have obviously found this very useful because we're able to gather up the liquid that is created when this reaction occurs in plants and in yeast, and then we drink it and it makes us drunk. Okay, so I hope that's helpful. That's everything that you need to know about anaerobic respiration. Well done.